Welcome to Fever Pattern, where every symptom has a microscopic source. In today's video, we're going to break down the gram-positive and gram-negative cell envelope structure, how they are built, and what makes them fundamentally different. The gram-positive cell envelope consists of an inner membrane surrounding the cytosol, with a massive peptidoglycan layer sitting directly on top of it. This peptidoglycan is the actual cell wall of the bacteria and the primary target for many antibiotics. Because it's completely exposed to the outside world, gram-positive bacteria need this wall to be incredibly thick and strong. Still, the exposed nature of the cell wall creates specific clinical vulnerabilities that we will cover in later episodes. Now let's look at the gram-negative cell envelope. The architecture here is completely different. The cytosol is still encircled by an inner membrane, but gram-negative bacteria also deploy a tough outer membrane. Their peptidoglycan layer is sandwiched right between these two lipid layers. It is much thinner than in gram-positives, but it's hidden and highly protected. We call this gap the periplasmic space. The outer membrane itself contains a large amount of lipopolysaccharide, or LPS, which is highly toxic to humans. It is also embedded with special proteins called porins, which strictly regulate what is allowed to enter the periplasmic space. So how does gram staining expose these differences in the lab? This method provides the rapid differentiation that is absolutely essential for our daily clinical work. After fixation, we flood the specimen with crystal violet, turning both gram-positives and gram-negatives a deep purple color. Next, iodine is added to reinforce the dye's connection with the cell wall. Then comes the critical third step, the alcohol wash. This doesn't affect the thick gram-positive wall, but it completely disrupts the gram-negative outer membrane, washing away the dye and decolorizing the cell. In the final step, a counter stain, like safranin, is added. This colors the now empty gram-negative envelope a bright pink, while the gram-positives remain solidly purple. While the gram stain is our fastest diagnostic tool, we need to know its limitations. There are certain bacteria that completely evade this test. The most famous of these are the mycobacteria, like the one that causes tuberculosis. These bacteria has a cell envelope heavily fortified with mycolic acids. This acid repels the gram stain entirely, forcing us to use a special acid-fast stain instead. And there are more. For example, mycoplasma has absolutely no cell wall, meaning there is nothing for the crystal violet to bind to. And certain pathogens, like treponema, are simply too thin or too strictly intracellular to be picked up on a standard slide. So if your patient is sick, but the gram stain comes back negative, be aware of this. If you want to decode more clinical microbiology and infectious diseases, please hit like and subscribe to the channel. Thanks for watching and see you next time.